Yeah, I'm Richard Anderson. I'm one of the editors of Rbmo, and it's my pleasure to join you this evening. This is our first live session this year, and we start with laboratory techniques, the technology underpinning our specialty. And this actually, the main speaker tonight is highlighting an article from our newest type of article in the journal Rbmo, and we've called that countercurrents contributions. So as you can guess, they're going to be controversial, contentious issues, but underpinned with rigorous scientific perspective. So it's a great pleasure this evening to be co-moderating it with Dr. Mina Alikani. Mina has fantastic years of experience. I hesitate to say how many, because she looks like she doesn't have them under her belt, but she does. She has decades of experience as a clinical embryologist. She's focused on improving IVF laboratory performance and outcomes through meticulous practice, detailed observation, exactly what the embryologist should be doing. And she has been a long-standing editor of Rbmo and lectured worldwide on this and many other related topics, published 80 articles, book chapters on laboratory aspects. So it's a pleasure to Mina for you to be with us tonight. So I'll leave it to you to kick off the session.
Thank you so much, Richard, and thank you for that lovely and kind introduction. I'd like to, in turn, introduce you and Marcos to our audience. Richard is, as he said, an editor of Rbmo, and I've had the pleasure of working with him since 2017. He's a professor of clinical reproductive science at the University of Edinburgh and works clinically in infertility and reproductive endocrinology, and provides a specialist endocrinology service to the Edinburgh Gender Identity Clinic. He has interests in both female and male fertility, with a long-standing work in the development of hormonal male contraception, very interesting areas of research. He has a major interest in fertility preservation and the effects of cancer treatments on fertility. And he has also conducted clinical studies developing our understanding of novel neuropeptides in human reproductive function in men and women. He's past coordinator of the ESHRE, a special interest group in fertility preservation, and is a member of the ESHRE Executive Committee and serves on the HFEA Scientific and Clinical Advances Advisory Committee. Of course, his full bio is available to you online if you would like to read more about his work and background. Together with Richard, I have the pleasure and honor of co-moderating the session with Marcos Meseguer, who really needs no introduction, except that he did remind me to say that, to mention that he is handsome and smart. So I am mentioning it, is that okay? That he's best known for being a scientific supervisor and senior embryologist in the IVF unit of Rbmo Valencia. But is also associate professor of the master's program in biotechnology at Valencia University. He's part of the group of researchers most cited in the world, with over 200 scientific publications, 70 review articles, and numerous book chapters. Marcus, it is a pleasure to be here with you, and as well you, Richard.
Thank you very much, Mina, for this introduction. Thank you, and thank you also, Richard, for being here today in this session. Well, it's my pleasure. I guess that is my time now. It's my pleasure to be here. It's an honor. I would like to congratulate also Rbmo Online for this initiative that I think is very, very, very interesting. It's a good way to bring the authors to the audience and have direct questions to them, which is amazing. Even today that we have a wonderful panel of speakers that we combine very well recognized to new young scientists that are coming, and we will see for sure for them papers in the next years. And again, it's my pleasure to introduce David Gardner, which is for me definitely the best embryologist of the world by far, well recognized, a very, even still very young, for me, embryologist, with the experience that he has, with all the publications, with all the reference that we have from him, just because every day we mention him when we talk about the embryo grading. His experiences here in Colorado, which made him very known about his research. Actually, he moved to Australia. And finally, we're going to have the chance to listen a good mixture of English between UK, in which he was born, US, he was working, and now Australia. A good combination of wonderful accents of English. He's going to talk about the way to improve RT using technology, and we are eager to listen to his presentation.
I'd like to start by thanking RBM Online for their kind invitation to talk today about the way to improve ART outcomes is to introduce more technologies in the laboratory, and of course to i3 for facilitating this event. Here are my disclosures. I get research funds from Virtus Health and Vitrolife, and I'm on the Scientific Advisory Committee for Fertilis, a startup company here in Australia. I'd also like to thank the many talented scientists who have contributed to this field over many years. And I sincerely apologize if I'm not able to cite all of your works in this very brief presentation. So this is really what we're trying to mimic in our laboratories and when we do IVF. And so much to talk about on just one slide. Lest we forget, one of the first parts of the female tract is to actually select the sperm that go on to fertilize the oocyte. And of course, we can see the dynamics of embryo development. The embryo, as you can see, undergoes morphological changes as it develops from the fertilized egg to the blastocyst, but it also undergoes dramatic changes in its physiology and metabolism, and therefore the nutrient requirements of the cleavage stage embryo are very different to that of the blastocyst. All the time the embryo is moving, it's not static. I mean, it does progress, but if you've ever looked at an oviduct, an excised oviduct after fertilization in a mouse, you can certainly see the embryo is moving around quite readily within the oviduct. Along the fallopian tube and into the uterus, there are gradients of oxygen, pH, cytocrines, growth factors, and indeed nutrients. And there's also an exchange of materials between the embryo and the maternal tract. So the point about that is this is not a static system, and it's about as far removed from our polystyrene culture dishes as one can imagine. So this is the countercurrent article that I was invited to contribute to RBM Online. And I would like to thank Bart Bowser, Laura Rienzi, and Mina Alikani for their kind invitation to write this. This is online now, it'll come out in the next month or so. I hope you enjoy reading it. It's a brief but insightful piece of work, I thought. I thought it would be prudent to define what is considered to be improved outcomes. And obviously the first thing that comes to mind is increased live birth rate, but it should also include the time to pregnancy, cumulative pregnancy rates per oocyte retrieval, and of course the health of the children. Furthermore, I would contest that anything that adds to improved management of our laboratories and contributes to the overall success of the clinic. So I think that's an important outcome too. So this is the table in the article itself, and these are the various technologies which I covered and I'll cover today, but very briefly, but I will cover each of them. And what you'll see is that they function not in isolation, but they actually work in synergy with each other, which is the really exciting part. But before one can look forward, you really do need to look back to see and appreciate how far we've come in the last 40 years. If I take embryo culture, something close to my heart, this is the kind of culture media that we were using at the start of our journey in human IVF. And indeed, Earl salts was one that Bob Edwards in particular used. As you can see, they're very simple salt solutions with added carbohydrates. There are no amino acids, no vitamins, And pretty much that's it. And that was pretty much the state of the art in embryo culture until the late 80s. And to make matters more interesting, we added patient serum. And we now know that serum is probably not one of the best things you can add to a culture environment for your embryo. And I show this for several reasons. This is a lovely picture of the founder editor of RBM Online, Bob Edwards, and Jean Purdy here, in an early laboratory. And really, what I want to focus on in this picture is the incubator. Now, this is a CO2 regulating incubator, but it was many, many years before we got incubators that could regulate oxygen as well. And what I would like to focus on is actually this part here. This is a glass desiccator, and this is what the team, Bob and his team, used to grow Louise Brown. It's a glass desiccator, and the desiccant's been removed, and you can place your embryo here. You could put a little bit of water in the bottom to humidify the chamber, but you can gas this chamber with a mixed gas if you want to. And indeed, at the very start of the 80s, on a trip to Braun Hall to meet Bob with Henry Lees, Bob showed us the desiccator, and I ended up doing the entire work of my PhD growing embryos in this system. It's very effective, but hardly high throughput. So let's look to the future. This is a review I penned nearly 30 years ago, and I was actually looking to the future back then, particularly with regards to culturing embryos in the absence of serum or co-culture. And in it, I was predicting what was going to happen. We already had the beginnings of sequential media. So I was talking about the introduction of blastocyst culture, indeed trophectoderm biopsy, the use of biomarkers, and all of which have come to fruition. However, one of the things I focused on has yet to come to fruition, and this was perfusion culture. So I alluded to the fact that the embryo in vivo is in a highly dynamic state, moving all the time, exposed to gradients. Could we recreate this in vitro? Could we introduce a changing environment to the embryo? And also, could we actually take the medium, what the embryo has been exposed to, and analyze that for biomarkers? So as I alluded to, this is something that we haven't seen come to fruition yet, but I think, as I'll show you, we're pretty close now to realizing this. Microfluidics, something we talk about a lot. It really is the behavior and manipulation of fluids in the sub-millimeter scales. So you're looking at fluids down in the nano and femtoliter rather than million microliter. And at such levels, the fluids flow by laminar streaming. They don't mix. And that makes them an exceptional process for separating cells, particularly in motile cells from motile cells, in this case sperm. And indeed, this is one of the very first applications of microfluidics. And like all really interesting things in our field, it's usually the marriage of a reproductive biologist and somebody outside our field. In this case, a macromolecular engineer in the name of Shu Takayama, working with Gary Smith in Ann Arbor, Michigan. And Shu and Gary were able to come up with this very elegant device whereby they could create a system with laminar flow streaming across the side. So at this interface, only motile cells can cross. And indeed, that's exactly what they showed, that as the fluid progressed, only motile cells were coming off the motile sperm, and they were able to collect motile sperm from that. So now we have commercial systems available for this already. And this was a paper from Jean Piero and Zev in Cornell, and where they were looking at specimens with high levels of sperm chromatin and fragmentation. And what they were able to show is using a microfluidic selection system, you could actually enhance the collection of sperm with high genomic integrity. And I think that's really important. So research in this area continues. This is a group led by Majid Warkiani in Sydney, here in Australia. And they've been developing microfluidic processes to, again, look at sperm, in this case, using sperm biopsies, and have shown that it's actually a very effective means of sorting out live sperm from a sperm or a testis biopsy. This is a really interesting development that Shu pioneered. And this is the use of a Braille system, pneumatic Braille system, to create fluid flow in a microfluidic device. And this is the microfluidic device here. It's a cast in PDMS, polydimethyl siloxane, which is gas permeable, and your embryos can sit in here, and the Braille system pumps media along. As proof of principle, Gary then went on to show that if you look at embryos in a microdrop or in a microfluidic system but static, the most important thing was you could get embryos to develop in a microfluidic device. But when you looked at a dynamic system, in other words, when you activated the Braille system to actually move the medium and therefore agitate the embryos, you actually saw a significant increase in embryo cell number. Now, that's not changing the media. That is actually changing the physical environment around the embryo. And one of the actions it's causing is probably breaking down unstirred layers around the embryo and making sure they can actually get access to the gases and the nutrients they need. We use microfluidics for the analysis of cell metabolism. This is a device we created with engineers at MIT and Todd Thurston's group in the mid-80s. And here we use pneumatic valving to pump small amounts of fluid. And this is a 3 nanoliter sample culture medium. This is a 10 nanoliter reagent chamber. And we introduced enzymes, and we measured the fluorescence in a reaction to quantitate glucose pyruvate and lactate. And it's a very elegant system, and it's driven by gas pneumatics, computer controlled. And here we can see flow rates of nanoliters per second. So several nanoliters per second have been pumped around this chip. Once it was all mixed, we could take our fluorescence measurement, quantitate the glucose, lactate pyruvate, empty out the chamber, and start again. So clearly a very powerful method of analyzing small amounts of fluid and moving it. But it hasn't led to perfusion culture as yet. And I think changes in microfabrication that we've seen over the last decade really offer great promise in this field. So using 3D printing and 2-photon polymerization lithography, we can create these incredible structures down at the micrometer level. And this has increasingly been used in biological scaffolds for 3D cell cultures. But here we see the Eiffel Tower. And if you just notice, that's 100 microns across. So our embryo would just sit right by the tower here. But you can see now we can get down to 1 micron resolution, this teapot at 1 micron resolution. It's quite incredible what can be done using this technology. So this approach has been used by my colleague Jeremy Thompson, who's pioneered a method to create a system for embryo perfusion. And again, using the 2-photon system, he's created a device that is transparent. It's not embryo toxic, it's consistent with embryo development, and it has microperfusion capacity built in. And this is the device. These are mouse embryos, and this is what has been referred to as the cradle. The embryos reside in each cradle, which then can be docked into a garage. And here we see the cradles docked within the garage. And each of these is built-in microperfusion capability. In this schematic, we can see the cradle itself, and these are the garages which have been inserted into this nest. And the nest itself can have culture media attached through tubes and then perfused across the embryo. And using this approach, we can get down to nanoliters of fluid per minute, which is a really exquisite control over the environment of the embryo. So the embryo itself is added into the cradle quite easily through a pipette, which then goes into the docking station or garage. Culture medium can then be passed over the embryos itself, more replicating the environment in vivo. It's dynamic. And this is what it looks like from above. These are mouse blastocysts placed within the cradle within the garage. And the pump is on, so fluid is coming through. And what I want to show you is when the pump is turned off, the embryos actually move. So by turning the pump on and off, the embryos agitate like this. And then you'll see this film of culture medium dyed red progress across the device. So the pump is on, and culture medium is coming through, pump off, and the embryos have moved back. When the pump is turned on again, watch the embryos move. There they go. And now you can see the culture medium perfusing across the embryos. You can also see that these outputs could readily have other connectors on, which could, as I'll show you, link to the analysis of biomarkers. What about microrobotics? This would be incredibly exciting for challenging techniques such as ICSI. And indeed, Yun Su has published 2 very detailed and very elegant papers on robotic ICSI. And this could indeed standardize how we do one of the most difficult processes in the IVF lab. Time release of embryotrophic factors. There's been much research on the addition of growth factors and cytokines over the years. And this is a recent paper that we published looking at a combination of growth factors and cytokines in the mouse embryo. And what we found was not only did these growth factors and cytokines stimulate mouse embryo development, but they improved development after transfer. But when we looked at the expression of genes in the fetus and placenta, we saw differences compared to in vivo. And our conclusion was that this combination of factors, although it had given us a benefit, was having a level at the gene expression level that we hadn't anticipated. Was this because we did a continuous exposure of the embryos to these factors, when indeed what we need to do is specific, stage-specific exposure to the factors? So we came up with the idea of creating a device that could actually deliver the growth factors in a time-wise state. And this is the first try we had. This is a synthetic blastocyst we created. This is made out of polyl-lactide. But in this case, in this pilot study, we used silica-based particles, and then we managed to embed them with VEGF. And Catherine, one of our grad students, was able to show quite clearly that when you added these slow release particles, you got the same effect as if the growth factor had just been added to the culture medium. So this means we could add these at different stages if we needed to. And indeed, Catherine then went on to use the polyl-lactide, the synthetic blastocyst as we call it, to look at interactions of signaling with the endometrium in vitro. So they could be very useful both clinically and indeed experimentally. I'd now like to turn to novel microscopies. Over recent years, both fluorescence, lifetime imaging microscopy, and hyperspectral imaging have been used experimentally. And now we're starting to see their use in diagnostics clinically. And this holds great potential for the field of IVF. This work has been pioneered out of Boston by Dan Needleman's group with Denny Sarkis. And the question is, can we use these kind of imaging to assess the metabolic state of embryos? Particularly, can we use these to look at key cofactors such as NAD, NADPH, and flavin, adenine dinucleotide, to quantitate the metabolic activity indirectly through light. And here we can see on the left, mouse embryos that have been excited, and we're looking at NAD levels, and I'll come back to the significance of that in a minute. A couple of years ago, I wrote an editorial on one of my own papers, which was on the grading system for the blastocyst and why the various parameters were included. And I thought it was important to note about aneuploid cells, talk about those, because aneuploid somatic cells have a phenomenon known as aneuploid stress, whereby an altered karyotype leads to altered gene expression, proteome, and metabolism. That's important to think about. So an aneuploid cell undergoes this process of aneuploid stress, and it's a phenotype of that cell. Can we quantitate that phenotype? And this was important because data today indicated that aneuploid embryos may take longer time from initiation of cavitation to full expansion. Does that relate to a compromised metabolic state? And this paper came out at the end of last year. Tiffany Tan, working in Kylie Dunning's lab, it's a very elegant study using hyperspectral analysis to look at the differences between aneuploid and euploid cells. The title says inner cell mass of the pre-implantation embryo. They were using mouse embryos, and they use chemical induction of large-scale aneuploidy. But they also looked at cell lines, and these are human embryonic fibroblasts. And you can see that when they looked at the NADPH, the flavin levels, and the redox, the ratios, there were differences between euploid and aneuploid embryos at each stage. Again, coming back to the fact that aneuploidy in somatic cells leads to aneuploid stress, and this is manifest as a change in metabolism. This month, this paper came out online from Daniel and Denny, again showing that FLIM could be used to detect differences, in this case, between euploid and aneuploid human blastocysts. So these are the blastocysts and these are the data. And when you look at the NADPH engaged fraction, the FAD intensity, and the redox ratio, there were significant differences between euploid and aneuploid embryos. What was really exciting about this work is that, of course, you can look through the microscope at differences between the inner cell mass and the trifectoderm. And when they looked at the inner cell mass, there were differences again in the NADPH, FAD, and redox. And in fact, these differences were greater than in the entire blastocyst. And then when the trifectoderm was considered, there were differences, certainly more in the NADPH, but the redox wasn't any different. So what this tells us is a couple of things, is that FLIM can differentiate euploid from aneuploid, and that not only can it differentiate the inner cell mass from the trifectoderm, but the trifectoderm is behaving differently to the inner cell mass, something that we know through detailed analysis by disaggregating embryos, but here we can do it visually. And this is a chapter that I wrote with Jeremy in Peter Nage's book, and it covers a lot of those things that I've just said in quite great detail. What about non-invasive biomarkers of viability in health? Here we have all the different stages of the human embryo as it develops and differentiates, and the ideal features. And this is from a review I penned with Bashak Balaban a few years ago, posing the question, is looking good still important in this era of all these technologies? And I would hold it is. It still is important to include this as a parameter. But beyond looking at morphology, what else can we do? And this is something a few labs around the world have invested in for many years. In fact, excuse me, I started my PhD work on this very subject with Henry many, many years ago. And we are able to measure the uptake of glucose pyruvate and some amino acids into individual embryos and quantitate the production of metabolites by the embryo. We could also look at DNA, microRNAs, exosomes, and that could eventually lead to non-invasive PGT. So by sampling the culture medium, we can actually get quite a lot of information about the human embryo. And as technologies advances, our confidence in the accuracy of these assays are increasing. But you may ask, why metabolism? What's the significance? Well, I've linked it to the fact that metabolism is fundamental to the survival of any cell. The pre-implantation embryo undergoes these dramatic shifts in metabolic function as it develops and differentiates. And what we know is that a lack of control in this transition leads to poor embryo development and a loss of viability. So it's a very useful biomarker. And indeed, these data from some 20 years ago show the metabolism of human blastocysts taken from the same patient. And these embryos were all of the same grade. And what I really just want to show you is that when you looked at the glucose uptakes here, there was over 100 picomoles in variation in activity between the highest glucose consumption and the lowest. And these were from the same patient and had the same alphanumeric grade. So you couldn't differentiate these embryos by looking at them. And yet metabolically, they were very different. This work then went on to look at glucose consumption of individual human embryos prior to transfer. And again, you can see that glucose uptake is related to pregnancy. So what we're saying is the higher metabolic activity, the higher the chance that that blastocyst will lead to a pregnancy. Now, this has been something that's been shown in the cow, it's been shown in the mouse, and it's been shown in the human. So metabolic activity is positively correlated to viability at the blastocyst stage. The more glucose consumed, the more likely you are to have a pregnancy. And this work took it to the next level. This was work by another graduate student, Laura. And Laura was looking not only at the relationship between metabolism and pregnancy, but all the other things that we were currently using, such as the KIDS score and AI ranking. Again, Laura concluded that glucose uptake was highest in those blastocysts that go on to give us a pregnancy. Interestingly, when you looked at the glucose uptake during the time of cavitation to full expansion, those that cavitate quickest and fully expand actually have a higher glucose uptake, which could be consistent with their karyotype, or it could be simply that they're using more energy to create the blastocyst quicker. We went on to show that glucose uptake was related to the grade of the embryo, the KIDS score, and even the artificial intelligence ranking that we gave it. And when we looked at amino acids, this is one graph taken from the paper where we looked at the AI scores, and those embryos with the lowest AI scores had a different pattern of amino acid consumption than those with a higher AI score. It wasn't just absolute more or less, it was a difference between consumption and production, which implies that, again, these embryos are fundamentally different in their physiologies. I just need to go back in time 20 years one more time, looking at glucose uptake on successive days of embryo development in the human. And the white bar is glucose uptake by human embryos on day 4, day 5, and day 6. And as you can see, as development increases, glucose uptake continues to increase. The reason for showing you this graph is that this paper came out last month, again with Denny and Daniel, showing exactly the same pattern, but this time with light. So whereas previously we've measured and quantitated the metabolites in the culture medium, this proof of principle paper is showing us that we can use FLIM microscopy to actually look at the metabolic activity of embryos, and it's consistent with our previous works. I think it's important to acknowledge that right now we're entering a paradigm shift in our understanding of development, and that's through this process known as metaboloepigenetics, first coined by Donahoe and Pultman a decade ago now. And in essence, it's relating metabolites and cofactors within the cell to gene expression. And Kevin Sinclair and his team in Nottingham have been working on one-carbon metabolism for many years and have shown the relationship with methylation status of histones. Remarkable work. More recently, it's become evident that NAD, which is an activator of sirtuins, histone deacetylases, can actually regulate gene expression. So therefore, the amount of this one compound can be a marker of the epigenetic state. And given the fact I showed you a few slides ago that you can quantitate this using FLIM, could we use FLIM to actually give us an indirect epigenetic status of our embryos? And now moving into artificial intelligence. This was the first paper I was very privileged to be part of, looking at deep learning for predicting of viability at blastocyst transfer. But AI has been used by many groups since then. And in these next two papers, I think what's really interesting is that both come to the same conclusions, that AI is very useful, but using a combination of both embryo evaluation, using the embryologists and artificial intelligence was the greatest benefit. And in the second paper here, I love the title of this, Should There Be an AI in Team? The same conclusion is the incorporation of an AI framework for blastocyst selection enhanced the performance of trained embryologists. So again, it's not that AI and humans are exclusive. In fact, people who work in AI have acknowledged the fact that actually you get the best results when AI and human intelligence is used in combination to make the final decision. And this amazing paper that came out quite recently was looking at sperm morphology and using deep learning for automatic assessment of selecting sperm. So this holds great promise now, not only using the velocity of sperm, etc., that's been used for many years, but to actually get down and look at strict criteria used for assessing sperm for selection. And this paper is really worth the read. This is work by Charlie Borman and Carol, who have really done some fascinating work in the area of AI and IVF. And in this one, they've used it to help us predict the parameters within the IVF lab that are affecting the functioning of the IVF lab. And I only wish that I'd had this when I was starting my career as a lab director. It would have helped me sleep so much better if I was having all this input into how my lab was working. And of course, beyond the IVF lab, Gerard Lettery has pioneered and discussed the use of AI to help physicians in creating predictive algorithms to help them do more optimal stimulations. So I'd like to try and bring all of this together for you in one slide. And this is a review that we penned a couple of years ago now. And this is the sort of focus of that review is we have come so far with these new technologies, and others are emerging rapidly, that rather than perceive them in isolation, look at the power, the synergies that they will have together. So in this schematic, I've got my microperfusion device, whereby my embryo is cultured, and I can change the input of media. I can change the nutrients over time. I can expose those embryos to growth factors and cytokines at stage-specific events. We could then take channels out of those ports and look for biomarkers, and then biomarkers could be metabolites, proteins, microRNAs, exosomes, and cell-free DNA. All of this can be imaged through time-lapse using either hyperspectral or FLIM. Now imagine the amount of data we're going to get from this. We've got image analysis data and we've got biomarker analysis. How can we possibly make sense of all of that information? Well, I think this is where AI will really come to fruition, to take all of that and create new algorithms for embryo health score. And I do say that deliberately, embryo health, because looking at all these different parameters, we're not just talking about the viable embryo, we should be able to distinguish the health of the embryo and therefore looking downstream into the health of the subsequent children born. So in summary, these new technologies, I propose, will increase the efficiency and effectiveness of IVF. Microfluidics is already a platform that's used for sperm diagnosis preparation, and advances are rapidly progressing. And I envisage that we will see more diagnostics on those chips as well in the near future. Microfabrication using 3D printing provides the opportunity to finally realize the potential of perfusion culture. We've only been waiting 30 odd years, but I think this is a very exciting time in the next few years for the full evaluations of these technologies. Microrobotics could indeed help us automate how we do ICSI. And I think FLIM and hyperspectral could provide new insights, not only into the metabolic status of the embryo, but also into the carrier type. Analysis of culture media has already been used to quantitate biomarkers such as glucose. But I think that with that and new microscopies, we're really looking at the role through the metabolo-epigenetic axis of having a chance of looking at the epigenetic status of our embryos. AI not only provides us great insight into gamete and embryo selection, but enhanced laboratory management. And I think this is really, really key when we get to this level of having so much information that we'll need AI to help us create algorithms because of the vast amounts of data we're collecting per embryo. And finally, and probably most importantly, I think what this clearly indicates is that less time is going to be used for some of the processes that we do in IVF, and we will make them more efficient. But I want to acknowledge that embryologists will always be required. And my take on this is, yes, it may take us less time to do various processes, and therefore it may take less embryologists to do 1,000 cycles per se. But these technologies will continue the success and growth of IVF, and therefore there'll be more cycles to do. Therefore, we still need the same number of embryologists. I think that's very important. These staff will be highly skilled and well educated. So I'd just like to thank you for your attention and thank my team, both at the university, the many grad students who've contributed significantly to our endeavors in this area, our clinical team at Melbourne IVF, and my collaborators for the last 30 odd years, particularly Jeremy and Denny, who have both been exceptional colleagues and very dear friends on this 30-year journey together. So thank you very much.
Thank you so much, David. That was a fantastic talk, and you're getting rave reviews as we as you're talking and as we are moving through your talk. Richard, would you like to ask David a question at this point? We just do a quick question.
Perhaps a quick one, David. Thank you very much for a really fascinating story. And I love the way you started with the real pioneers in the field. It always means so much when we recognize where we came from and what they built and their legacy. I think that's just lovely to see those photographs. But it's I guess so sorry?
So important. I absolutely agree.
It is.
You know, we're only standing on the shoulders of giants. That's the reality.
Absolutely. Huge giants in those particular cases. I guess my question here is that the techniques that you've talked about today, microfluidics, the microscopy, are all really exciting, but they're up against the challenge of the biology here. And you alluded to it, particularly in your comments on the glucose uptake, which varies enormously between otherwise, undistinguishable but competent embryos. And that's always seems to be the challenge with these new technologies in embryology, is you can show group differences, but translating that into a diagnostic test for clinical use when you need really clear differences between groups is always more of a challenge. And so it always seems to me that these are so much better in the research lab than in the clinical environments. So what would your thoughts on that?
Yeah, I mean, that's a very, very poignant and relative observation, Richard. The reality is, those prospective trials really still need to be done on those biomarkers, and that's something that we are planning to do. But I think you're right with regards to this overlap. I mean, people are concerned when you read the papers, they get very excited, and the Flynn work is exceptional in that regard. But you can see there's overlaps on those box plots, for example. The same of some of the glucose data, although it's quite a degree of separation, there is some overlap. The way I tried to draw it together on that last slide is to say, well, perhaps if we just focus on one thing, then we're not going to have the full prognostic potential. But if we put all these things together, I think then we're in the real driver's seat to actually have a greater assurance that we are picking the healthiest embryo, the euploid embryo, and then we can rank them in that way. So I really do think it's going to be a combination of technologies. Don't look at one thing, see them all as complementary. And because they're all asking quite different but similar questions. And I think that's what's exciting.
Just as you would never buy a house just on the color of the carpet in the living room alone, it would be that you need to see the other rooms as well.
Exactly, yeah.
Thanks for a lovely talk.
You're welcome, thank you.
Thank you, Richard, and thank you, David, again. So we'll come back to you during the Q&A session later. At this time, I'm really excited to introduce this session's speed round. And for today, we have 3 speakers who will present their very interesting work that we published recently in Rbmo. And we start with Anna Pereira Daoud, who will give a talk to us with the intriguing title of The Closer the Knit, the Tighter the Fit, Conceptual and Ethical Issues of Human Embryo Modeling. Anna received her master's degree in applied ethics in 2018 and has since been pursuing a PhD in the Netherlands at Maastricht University. Her research focuses on investigating the ethical ramifications of research with 3D human embryo-like structures and aims to contribute to the development of tangible guidelines for harnessing the potential of this valuable research tool. So without further ado, Anna, please go ahead.
Thank you so much, Mina, for the kind introduction, and good afternoon or good evening in my case to everyone. I'd like to begin by saying thanks, of course, to RBM Online and IVF Initiative for inviting me to speak at this exciting event, and to everyone that's watching online for joining us in it. All right, I can move on to the next slide. But before that, I will tell you a little bit more about myself, not that you need to know after the great introduction by Mina, but my name is Anna once again. And as I'm sure you'll notice, I have a little bit of a different background than the rest of the amazing speakers here tonight. I am an ethicist and I am in the last year of my PhD project at Maastricht University, where I research indeed the ethical ramifications of 3D stem cell-based human embryo models, or as I prefer to call them, human embryo-like structures. This project was funded by the University Medical Centers of Maastricht University and Utrecht University, the Merlin Institute, and the Dutch Organizations for Health Research and Development in the Netherlands. I do not have any conflicts of interest to declare. I do, however, have a lot to tell you. So in the following few minutes, I will briefly talk you through the highlights of the viewpoint our team published in Rbmo in last year, following the first-time creations of human blastoids. Let us begin. As you probably know, human blastoids are integrated embryo-like structures cultured from human pluripotent stem cells that resemble both the embryonic and the extraembryonic tissues typically found in blastocysts. There are many more types of embryo models, not all of which are integrated, and they can be scientifically useful in many different ways, but I will not focus on all of them here, as that is also not my expertise. What I will focus on, however, is what makes them specifically interesting from a political and ethical point of view, namely that they can enable, or at least seem to be able to enable, the benefits of human embryo research while avoiding the burdens associated with their research use, a topic that remains politically and societally divisive to this day. It is therefore not surprising that research with these models is widely regarded as a win-win policy, and therefore also attracting a great deal of governmental funding in, for example, the Netherlands. Now, this is of course assuming that human embryo-like structures are indeed models and not embryos. And it seems that that assumption is so far correct. Even the more refined human embryo-like structures have only a very limited capacity to model human embryogenesis, the improvement of which would, by the way, also require parallel human embryo research. So that should dim down immediate benefits of the aspired win-win policy. Should these hurdles be overcome, then it may become increasingly difficult to distinguish between human embryo-like structures that are just models and those that should be regarded as stem cell-derived embryos. There's a paradox that emerges here, in that the better that these models become at replacing human embryos in research, the less useful they may be as embryo-replacing models. Note that this is not because doing research with perfect replicas would be ethically problematic in itself, but rather because creating perfect replicas would place them back under the regulatory frameworks of human embryo research, which these models initially meant to circumvent. This brings me to the next slide on how a lack of a universal embryo definition complicates these matters further. Of course, whether or not embryo models qualify as embryos will have important consequences for the permissibility and the scope of their research use. The problem is that embryo definitions vary quite extensively across jurisdictions. In Spain, for example, the human embryo is defined as a product of fertilization. In the Netherlands, as a cell or cluster of cells with the potential to develop into a human being. For researchers working with these models, disparities in embryo definitions means that research with some types of embryo models will be severely limited in some jurisdictions, while not requiring the same or even any level of regulation in others, as is, for example, now the case with the human blastoids, blastoids created in Australia. For politicians, it means that there may be a way to have it both ways and avoid rather than address the ethical and legal questions raised by novel scientific developments such as these. Last but not least, in the last minute that I have, I would still like to tell you a little bit about the ethical issues, namely that these models are also sparking discussion about a corner argument in the traditional human embryo debate. This argument states that the human embryo deserves special moral treatment because unlike any other human or non-human cell, it has the potential to develop into a person like you and I. Supposing that this argument withstands, it is currently debated, but let us do it for the sake of the argument. Two side notes are still worth making. One, it is perfectly defensible to argue that human embryos can only have limited moral status on this basis, and that their research use is therefore allowed under conditions of proportionality and subsidiarity, which would also apply to embryo-like structures if they turn out to have the same potential as human embryos. 2, many authors have argued that the embryo's potential can only matter morally at post-individuation stages, meaning that only post-implantation stage embryos or embryo-like structures would qualify for protection on this basis, and blastoids or blastocysts would not. Other ethical issues can also be expected precisely with regard to the models that are clearly not embryos, and that for that reason are not bound to the restrictions imposed on human embryo research. But in view of time, I would like to keep it at this and kindly invite you instead to read the full argument online. Thank you, and please feel free to reach out with any questions.
Thank you so much, Anna. I also would like to encourage everyone to go to Rbmo and read your paper, which I find to be very, very interesting. I'm not sure how relevant this question is, but something that has come to my mind, and I wonder if you could shed some light on this. You mentioned political opportunism in your slide, and that caught my attention. As a political junkie. You know, in the US, the politics of embryo research are linked very much to the politics of abortion. And the concept or the premise that life begins at conception. And anything that resembles a human embryo or a human life has to be treated in a special way. And for that reason, no research is allowed with government funding in the United States. So how do you think we can ever address this and get over it?
Yeah, well, Mina, I have to say this is such an interesting question, and I'm happy you're a political junkie, as I am myself. I have to say, I don't know if it's gonna be possible, but I do feel like novel developments such as these definitely make it necessary. If I look particularly at the case in the Netherlands, which you described for the UK, it's also true here with this to a certain extent. For example, in the Netherlands, it is not allowed to create human embryos specifically for research, which means that human embryo research is limited to supernumerary embryos. That is a particular political stance, also based on that special respect due human embryos, and that is often, at least here in the Netherlands, grounded on that special potential that they have. There are two things I want to say about that in this particular context. First of all, again, there is debate about that potential. Is it that special? What if these models that we create from any human cell turn out to be able to acquire the same potential? That would potentially undermine the special status of human embryos. That is one side of the coin. And the other side of the coin as well, you can also just preemptively adapt these models so that you know that they can never reach that potential.
Right.
Which would be a little bit of a loophole, at least ethically speaking, right? It would meet the legal requirements, meaning that if human embryo-like structures have that potential, then you cannot create them in the Netherlands for research because they would qualify under the Dutch Embryos Act. So the alternative is to manipulate them and turn that off and find a loophole. In either case, it's a bit of a problematic situation. And this is exactly the reason I think that we really have to try to at least get one step further on this. A little bit more agreement would be nice.
Thank you so much again. And we'll come back to the discussion at the end during the Q&A session. Thanks again, Anna, and congratulations on your publication. Now it's my pleasure to introduce our next speaker, Romualdo Sciorio. And Romualdo received his first master's degree in biology in 1998 at the University of Naples, and later obtained a second master's degree in biotechnology at Edinburgh University. He worked between London, Dubai, Philippines, Armenia, Switzerland, and India as consultant embryologist. So he is obviously quite well traveled. before returning to Scotland in 2014 and Edinburgh, where he works as a senior clinical embryologist now. Now, let me see here. The topic of Romualdo's talk is focused on time-lapse analysis, blastocyst collapse, and morphometric assessment as new features of embryo viability. Romualdo, please.
Thank you. Thanks very much, Mina, for your introduction. And I'd like also to say thanks for the kind invitation to Rbmo Online. And it's a great pleasure for me to talk tonight about this recent published paper that, as I said, is a title is a focus on time-lapse analysis, blastocyst collapse, and morphometric assessment as a new future of embryo viability. The next slide, please. Well, basically, the main point of this review paper that we published with Meseguer was to highlight some benefit of time-lapse technology. As we know, time-lapse give the embryologists the possibility to observe, to recognize some specific phenotypes that otherwise with the standard culture will be never seen, such as abnormal cleavages, reverse cleavages, multinucleation, and even blastocyst collapse event, for example. And in addition, time-lapse provide a steady culture condition, which is really critical. Avoid to expose to embryo to a physiological environment. And as David before said, this is quite important because there is no changing in temperature, in oxygen concentration, CO2. So this is important to avoid those stress to the embryo that can impact embryo development. But the two main things I'd like to discuss tonight is related, as I said, with blastocyst collapse event and morphometric blastocyst assessment, where we take a measurement of blastocyst diameter and the blastocyst area, and we try to correlate those numbers with the pregnancy outcome. So in the next slide, I want to show the collapse event. So the blastocyst collapse event in humans was discussed for the first time by this paper by the group of Meseguer in 2015, and the authors report a reduction in implantation rate if the blastocyst has a collapse event during the development. And we were inspired by this paper, and in the next slide I'll show what we did an analysis in our data, and next slide, please. We basically reported the first paper, which was released in 2019, in which we took analysis about 350 blastocysts, and we see actually the same trend. So if the blastocyst has a collapse event, has a reduced of implantation rate, and has a reduced ongoing pregnancy rate. And this was statistically significant. The pictures in the center here show what it means for blastocyst collapse. As you can see, the embryo on the right is completely collapsed, as a reduction of the volume, which is more than 50%, compared to the embryo on the left, which is the same embryo just 5 minutes before. And this observation is normally done before 115 and 16 hours after insemination. So the table of the bottom is another study which we published in 2020, and this is a multi-center study. Included almost 1,500 blastocysts, and this was performed for 4 different units. And again, we see the same similar trend. We have, if a blastocyst has a collapse event and is transferred, as a reduction in pregnancy, a reduction in ongoing pregnancy rate. And here we also seen that the collapse group has an increase in miscarriage rate. So it's some thinking about genetic abnormality or eventually any aneuploidy in those embryos. So according with those studies and with other studies previously published like Bori, Meseguer, and many others in mouse and cow model, we can predict that the blastocyst collapse can be a negative markers of embryo implantation. And in the next slide, finally, I like to discuss what is the morphometric blastocyst assessment. And the question is blastocyst diameter and blastocyst area are correlated with the pregnancy outcome? And well, in our data, basically, we take a measurement of blastocyst diameter and we correlate this with pregnancy. So basically, as we see from the graph, the lady they are pregnant, they get a fetal heartbeat, they have an average a larger diameter, which is an average around 185 micrometers, compared with the lady which are not pregnant, and in this case, the diameter is around 160 micrometers. Same trend is for blastocyst area. So of course, these are retrospective study. And after a single embryo transfer, we'll see that basically the area, the surface of the blastocyst in a lady, pregnant lady, so lady that gets a fetal heartbeat, this is around 28,000 square micron, compared to the lady which are not pregnant and who have in average a small, let's say, surface of the blastocyst, which is around 22,000 square microns. So these observations, these parameters, they are numbers, so they are very objective. There is no variability between embryologists, so they can be easily applied and used in a scenario where we have 4 or 5 blastocysts to select for transfer, and of course you don't want to transfer a pool to avoid the increase of multiple pregnancy. So these parameters can be really taken in consideration to select the single blastocyst with high implantation potential for transfer. And in the next slide, which is actually my last slide, is just the picture of our paper, which is recently published in Rbmo online with Marcus Meseguer. And that's it. Thanks very much for your attention.
Thank you so much, Romualdo. That was a very interesting talk, and you described some really interesting observations. Again, our audience would benefit from reading your review in Rbmo. I wanted to ask you a quick question and see if you can speculate about why blastocyst collapse could be a marker of lower implantation potential, because after all, this may be a natural phenomenon that prepares the embryo for hatching. So why a lower implantation potential? And I also wanted to know whether you have made the same observations with embryos that have been exposed to assisted hatching.
Well, there are some hypotheses coming from more animal models, mainly in cow or mainly in mouse. In the humans, actually, the phenomenon is still unknown. But in a few studies performed in mouse model, they've been reported that after this blastocyst collapse, there is some kind of damage to gap junction and tight junction that they keep the cells, especially trophector cells, keeping together. In fact, there is a nice study from Japan, Tagashi, tried to use some inhibitor on gap junction. They use heptanol and oleamide as inhibitor of gap junction, and he reported, but again in the mouse model, that those blastocysts growing with the inhibitors of gap junction, they have a different collapse, and they are not really able to expand and to hatch, and then eventually they get to degeneration. So the problem, it could be related to these junction problems. And in addition, we need to take in consideration that every time the embryo is expanding, it's going to use sodium potassium pump. So there is a lot of energy which is involved in this process. So if the embryo, because some embryo, they do just a bit of contraction, which is slowly. So that one, according again with mouse model, if these are a small contraction, it will be helpful for the hatching. But if it's a strong collapse, like, you know, more than 50% of the volume, maybe the embryo will damage some junction and then has no energy to re-expand. So that would be the negative point. But again, a lot of these studies are on mouse model. It's quite difficult for, you know, the same topic we just discussed with the previous speaker, and it's almost impossible to study, to investigate this phenomenon on the human blastocyst, basically.
Yeah, very interesting. I hope we can come back to this later on in the session. Thank you again, and congratulations on your publication. Now we move on to our third speaker in this speed session. That is Lorena Bori Arnal. And Lorena is a PhD candidate researcher at Rbmo Global in Valencia, under the supervision of Dr. Marcos Meseguer, our co-moderator here. She received her biology degree in 2016 and her master's degree in biotechnology of human-assisted reproduction in 2018 at the University of Valencia in Spain. The primary area of her research interest is embryo evaluation and selection with non-invasive methodologies, especially through artificial intelligence. We are happy that she's here today to talk to us about a topic that matters a lot to all of us embryologists, and that is, will the introduction of automated art laboratory systems render the majority of embryologists redundant? Hopefully the answer is no, Lorena. Right. Please go ahead.
Okay.
Thank you.
Hello, everyone, and thank you very much, Mina, for the introduction. I will briefly present our countercurrent contribution on whether automatism will render the majority of embryologists redundant. Please, the next slide. Over the last 40 years, laboratory methodologies have been rapidly updated, including ICSI, embryo culture extension, vitrification, biopsy, or time-lapse systems. And currently, we are seeing the incorporation of automation in some routine processes. The automation will enter the embryology laboratory along with microfluidic technology for sperm isolation and microrobotic immobilization for ICSI. Also, the future sperm assessment would analyze the risk of fertilization failure and risk to offspring by methylation or mutation loads. In the same way, robotics, computerized systems, and artificial intelligence integrated with image analysis and biochemical evaluation of the oocyte quality could guide patient treatment decisions and automate some processes such as oocyte insemination by IVF or ICSI. Even intelligent systems for assessment of fertilization can be developed by means of image capture and three-dimensional reconstruction. In the next slide, we can see that, as we all know, that embryos in vivo are exposed to dynamic environments that provide a specific nutrient at each stage of development. But currently, in vitro embryo culture is based on static conditions, and future new techniques based on automatic devices with microfluidics can be used to regulate the medium flux, and probably the dispute between single or sequential media would end with the addition of biosensors to these devices for adapting the needs of its embryo. Sorry, but I think that the slide is not working. I don't know if you can click again. Yeah, thank you very much, Giles. Okay, so even the automation can be used for vitrification as the last step of the automatic device based on microfluidics. Also, the spent culture medium can be analyzed to assess the metabolic state of the embryo in terms of, for example, the oxidative processes or proteomic profiles that have been correlated with clinical outcomes, or even for non-invasive PGTA. Also, time-lapse images obtained during the embryo development can be used as input data for systems based on computer vision. I mean, a complex artificial intelligence system based on a combination of microfluidics, biomarkers of embryo metabolism, image analysis, and non-invasive PGTA could serve to create an embryo ranking for each cohort. And in the next slide, okay, we can see that there's no doubt that automation is entering embryology, as revealed by the increasing publications about artificial intelligence in IVF laboratories, as you can see on the graph. And nowadays, we have introduced the automation in some routine processes, such as electronic witnessing systems or for embryo assessment. And it's expected that in the near future, the similar automated systems will be introduced. For example, data collection can be performed automatically by voice recognition. And also facial recognition systems can be installed at workstations that would record automatically the date, time, process, and operators. Even the preparation of culture dishes and sampling medium for quality control can be automated. In this way, the key performance indicators will become increasingly complex, and systems based on big data can be used as an early warning to detect changes in operators and equipment performance. Therefore, the artificial intelligence will be introduced in several clinical processes such as embryo assessment, oocyte denudation, or fertilization checks. However, more complex procedures such as ICSI, trophectoderm biopsy, or vitrification may not be fully automated in the short term due to the highly technical and variable nature. Next slide, please. So it's inevitable that the implementation of automated and rapid systems will reduce the future demand from embryologists. However, when conventional tasks will be automated, other novel tasks will be created around the increasing amount of data that will be generated per embryo. The embryology would be much more technological, focused on advising computer scientists and engineers on the development of devices for embryo manipulation. Also, the new role of embryologists will involve exploring the data and determine its relevance, deciding how to use the huge amount of data and how we can combine it to obtain an accurate diagnostic of the competence of an embryo. Also, the embryologist must be able to broadcast what's in action in the laboratory to make this information accessible to a growing demand of patients who want to understand all the procedures occurring in this black box between the oocyte retrieval and the embryo transfer. Also, the new task that embryologists will face will require additional knowledge in statistic and database management. So in conclusion, also the implementation of automated systems will reduce the time spent by embryologists on their current tasks, and they will perform new functions. And in my opinion, the the expertise of embryologists will always be necessary to ensure the successful implementation of any new technology. And that's it. Thank you very much for listening.
Thank you so much, Lorena, and congratulations on your paper. Very interesting. Again, I have a question, but slightly tangential to what you discussed. You list QC/QA under a new role for embryologists, or remaining roles, let's say, for embryologists to play after automation, full automation. Currently QC/QA is a major part of our day in the lab, and we measure and remeasure, temperatures, gases, liquid nitrogen levels, and et cetera, et cetera. Do you think we could do better with more automation in that area as well, and could actually rely on our instruments to make redundant measurements so we don't have to constantly go in and manually measure what the instrument has already measured on its own? This is an area that I don't think has been addressed, and I wonder if you have any thoughts on it.
This is a very interesting question. I think that the best way to implement the automation for quality control is to teach a system based on big data. The system can be trained from data for different laboratories. So I think that the best way is to have a system that will be able to detect changes in our daily routine, changes in the temperature, in any any factor that can do that our outcomes will be worse. So I think that the best way to implement the automation in this way is for early warning, and to detect changes in the equipment performance, and be able to change the problem and become that the results will be better.
Thank you. Yeah, that's a very good comment and observation. So thanks again for a very nice talk, and congratulations. I'd like to invite all the panelists and speakers to the QA session, and we can officially begin the QA.
Yes, we're ready for the questions, and I guess that the first questions are going through David Gardner, and as far as I've seen, I think that Mina has the interest to make some questions to David. So I don't know if you want to start with the questions to him, or we just take a look to.
Yeah. So I saw a couple of questions that had come in from the audience, and I wanted to sort of pick up on that. Pose it to David, and then I wanted to add a couple of things to this topic area. So the question was, we have been speaking about microfluidics for over a decade. What has changed now to make this more applicable? And then the second question, which in my mind it's related, is the time, interval between initial introduction of a technology or science and actual clinical application. And this person then says, I'm sorry, I don't have your name, but what is your prediction for IVF in 2042, specifically, David?
That's a
2042.
great number. I often think about 2042 myself. It's a number to
Yeah.
behold.
I don't know how they came up with that year, but you know, it's a good year.
It's a good year.
2042.
A couple of decades to go. Yeah, I hope we're still around to play. So to go to your first question on the microfluidics, I mean, really, it has been around 2 decades. It's been around a long time. And I think what's been really exciting is that we've learned really what we can do with that technology. And I think from my perspective, it's really showing great advances in things like sperm selection, and the next generation of chips keeps coming, which is really promising. I can see those chips having diagnostics built into them. And they're really suited for those kinds of procedures. So I think we're going to see microfluidics reach its potential in more of a diagnostics role. Where it hasn't delivered, and there was lots of reasons for that, is in the embryo culture side of things. And I think what's really exciting for me is sort of the microfabrication story that I've been working with Jeremy Thompson for so many years, but really in the last few years using 3D printing to create devices that are more embryo friendly, more embryologist friendly to use. So I actually do think we are seeing, we're going to see more of microfluidics, but more in a diagnostics capacity. And I think microperfusion, which is not a microfluidic platform at all, and has much tighter control of the volume regulation and flow, will be potentially a way we can actually finally get around to that perfusion culture we've been talking about for over 30 years and really see if that's really going to live up to its potential, as Lorena was talking about too. You know, we've all been thinking about this, and I think we're very close to getting there. So that was answer to the first question. Happy for anyone else to chip in on that.
Thank you. Thank you, Mina. Just now, but first of all, I would like to congratulate David for his presentation. Amazing. It's always our big professor for all of us. And I'm just because I have the chance to have you here, and we follow the questions. I'm just thinking about the sequential and the single-step medium, and link to microfluidics, and just share the experience of many of us when we did the transition to single step, and we were very happy because it reduced the amount of work that we have in the lab. But then we start to realize that with some of our culture systems, we start to have some, some difference related with the culture conditions. We start to try, for example, in the single step using or not humidity, and we start to see like better outcome in those cycles with single step and using humidity compared to dry incubators. So let's say that we start to think, okay, if I use dry incubators, it's better to use sequential media. If I use humid incubators, I use single step. That's one thing. The other is that if we create a better environment for the embryos, let's say that we are less restrictive in their development. So let's say that also we select worst. We can have, that's my impression of the result I have checked, for example, with or without humidity. I have initially better outcome, but then I have more abortions. Let's say, so I have give more chance to the worst embryos to grow, but then I have at the end the same outcome. What do you think about that?
Okay, well, I'm not as concerned about the humidity and dry, because if you look at your system and check your osmolalities and pHs, you can make sure that you don't have differences, even in a dry system. So I've not been concerned about that per se. What I am concerned about, though, Marcus, is leaving an embryo in a drop for a really long time. And I think we've got away with it when we go from day 1 to day 5, for example. But the human embryo is, by day 4 to day 5, is metabolically incredibly active. You've got to remember the blastocyst is the most active tissue in the human body. It's got a higher O2 uptake than cardiac muscle. And it produces this acidic environment around itself. It also is very active in transamination. So it's pouring out ammonium, it's pouring out lactate, and its nutrient requirements are going up exponentially. And you could see from that graph, both from Denny's work and my work from 20 years ago, from day 4 to day 5 to day 6, there's a huge difference in glucose consumptions. And what that really means is that when you go from day 5, we have always renewed the medium, no matter what we've grown it in, but you must renew the medium on day 5 going to day 6, because that's a great example of how the embryo is changing its environment dramatically over time. So I've always felt safer, you know, having a sequential system was great in a smaller volume program where you had a humidicrib and you could take the time to move the embryos around and you didn't worry so much about pH and temperature shifts. But as clinics have got bigger and bigger and bigger, and we've used time-lapse, I can see the attraction of moving to a monophasic system. But I would still hold there are problems with that. And that really comes at the latter end, when the blastocyst becomes so active that it is completely changing its environment, and we have to renew it. So then it comes full circle to perfusion culture, not to be confused with microfluidics, but some way of changing the medium in situ, which I think, you know, Jeremy Thompson has shown that you can do. And so that I think actually holds promise that you could actually do perfusion culture in a time-lapse system. So why not have the best of both worlds? And I think, you know, we can speculate what's going to happen, and I'm, you know, quietly confident in what I predict would happen. But we have to do those studies yet. But I think that's a really fascinating part of the future. Mina, I haven't forgotten you, 2042.
Yes, yeah.
I mean, really, I think, you know, everybody's covered so many facets of what's going to happen. I mean, if you consider the oocyte journey from collection to transfer, I mean, what we're going to see is, in terms of some of the developments in optics, for example, we're going to have the capacity to actually measure the quality of an egg as it passes through in the aspiration tube. So as we aspirate it, we're going to get some feedback onto the status of the egg. And I think that is, you know, holding great promise for the future. All the things that we talked about, automations, I mean, they could well come to fruition, as could all the microscopies. But I think really the other side of it is, you know, we're going to see all this. I know we're going to talk about the lab, but if we can use artificial intelligence to help us get better stimulations and better endometrial receptivity through various tests and analysis, I think that's a huge part of what we do as well. So I think really just to go back through all of the things that we said between us, these things are sequentially going to be introduced over time. So by 2042, I think, well, at least half of those things that we talked about will have come to fruition, at least half. The question was about time from initial introduction to clinical use. This is a great question, and I've got to share my own story. And it was a simple one. It was called blastocyst culture. And what it required was 2 media and going from day 3 to day 5. And I thought, as did Bill Schoolcraft and the others, we thought that was a home run. How easy is this? And, you know, our pregnancy rates were going to go up. And all we were asking is to go from 1 media to 2 media in 48 hours. That was a 10-year battle to get people to do that. So we are up against it. I know people love toys, love technology, but the one thing I learned from just introducing 2 media from 1 is that these things take time. You have to be patient. You have to ensure that you've done enough pilot studies in appropriate models, and then do your prospective randomized trials. I think evidence-based medicine is the only way forward to introduce these things. Otherwise, we're cursed to reinvent history. And a lot of things over the last 20 years have come and gone and never really delivered. I think what we've all talked about today have got the potential to deliver. We just have to be patient in how we deliver it.
So you've provided the perfect segue to what I wanted to say next, which is share this analysis that Jacques Cohen did on predicting the future using time intervals from basic discovery to clinical application. And he put together a table, which is quite interesting. And he listed a few of the technologies. For example, IVF, animal models, 1959, clinical application, 1978, interval is 19 years, follicular stimulation, 1927, 1960, 33 years, vitrification, 1985, 2010, 25 years. So he listed all of these very important technologies in ART and came up with an interval, an average interval of 18.6 years for a technology or a discovery to make it to clinical application, and the range is 2 to 33 years.
Yeah,
So I think that's very
really
instructive.
interesting. Yeah, I think that's really interesting. What that table doesn't take into account are species differences. So although we could do a lot of those things in animal models, we couldn't do it in the human, not because it wasn't a translation, it's because we hadn't worked out how to do it.
Whereas well, exactly, that's why it's such a wide range.
But if you take a technology, like something automated, I think that's got a faster time than something that we have to wait.
You have a point there.
So in a way, it's still discriminating between biological or scientific breakthroughs as opposed to technological ones. Mm-hmm. And I think they're fundamentally different in their time frame. I don't disagree with Jack at all. But I think that perhaps that table could be redone in 2 columns. One that says scientific translation, and the one is technological translation, because as I alluded to, those are very different.
Well, in practice, it's still proving. But you said it's still proving, like microfluidics is a perfect example of it. It's technology, but, we don't have it yet. And it's been a couple of decades.
Well, in a way we do, though. I mean, there are commercial devices for sperm, but I think that's possibly where it's going to end, in sperm. Yeah. I'm not sure, I'm convinced yet that microfluidics is going to see its way into oocyte biology or even embryology. So it might have actually found its niche and doing very well, and it's going to grow in that area. I've yet to be convinced that microfluidics is the way to culture an embryo yet. But microperfusion could be, which is fundamentally different to microfluidics. Right. Because we're talking about microfluidics is the flow of submicroliter volumes, and you've got a laminar flow and all the rest of it that comes together. And you maximize the physics to do cell separation, as opposed to microperfusion, which can be turbulent flow, but you're getting very small volumes, and you can actually manipulate in exquisite detail what you do. So just, yeah.
Can I jump in and go back to the question list that we've been given by our audience? It's a fascinating conversation, but maybe a broader, more philosophical question, and this is addressed particularly to Anna, actually. Someone is asking, will these technologies, both yours and more mechanical innovations, be limited to more affluent countries? And we had a similar question from one of our audience from Ethiopia as well. So I think this is a really important question of spreading the knowledge across the world rather than just particular rich centers of excellence.
Yeah, thank you for that question, Richard. And indeed, sorry to interrupt, this was already a very interesting conversation. Obviously, this is not my topic per se, but I can give my 2 cents on the topic. I think that it's hard not to affect less affluent countries, in the sense that I do think that one of the benefits of these embryo models is that they are probably much easier to produce. You can produce them in great quantities. Perhaps in that sense, it might be more facilitated than traditional human embryo research is. But I'm not familiar with how it is monetarily, what the kind of costs that it would be involved in this kind of research. I don't know, perhaps the other panelists have a better idea of that. But my personal intuition would be that yes, ultimately these technologies will be more limited to affluent countries, as are more recent innovations in general. And actually, that is an intuition. Please prove me wrong. I would love to be proved wrong.
It's a really interesting point, and it actually goes with my camera's just gone.
I can still hear you.
Hang on. Okay. Well, so my other question related to that actually was perhaps to a number of the panelists, is to think about, we've talked about the advent of, there's a lot of technology involved there. And I think, David, you mentioned the embryologists, they like their equipment. Does this then just focus more and more the research and therefore the direction of travel in those who have the biggest budgets? And of course, then we have commercial interests who are able to fund this sort of thing, are obviously going to be promoting their own technology and perhaps steamroll of that into practice if at all possible.
Yeah. Mind if I have a first crack at that, Richard?
Well, have a shot, yeah.
Okay, yeah. One of the things that came up, and one of the questions was, how can developing countries afford such technologies, and are they going to be disadvantaged in this? And that could be a fair perception, but in the article I wrote in Rbmo, there's one little paragraph I wrote that talked about the fact that, okay, the whole concept of what we're talking about is advanced technologies, But with someone like Mina, working with a really great physician, Mina, you could hold your own if you only had to do 200 cycles a year, you know, and you could get great results. You don't need the full Monty that we're talking about. I hope you would agree. I mean, you can get great pregnancy rates working just with basic approach to embryology, i.e. just as we used to do it. But it's unrealistic to be able to do that as the volumes grow and grow. I mean, programs are huge these days, or have the capacity to be huge. And the concept of an artisan embryologist is almost non-existent now. And I think, you know, Marcus, you used that term as well when one of your, and I think it's a great term, an artisan, because the boomers still, you know, consider ourselves artisans. Yeah, I remember growing my embryos in those glass desiccators. I mean, you know, we made it up as we went along, and we did really well. But the technology, so what I'm saying from that is that if you don't have these technologies, if you have skilled people, you can still get very good clinical results. That's my premise.
You have the basic knowledge and understanding.
Yeah, technology understanding just makes it, you know, it leads the way for efficiencies, improvements, time saving, those kind of things. Yeah. So yeah, I think you know, that's how I perceive it.
Thinking also in making universal, I mean, the IVF universal and the access to IVF to the all around the world, because one of the main problems in many countries, is not the case of Australia, is that some of the patients cannot afford the price of the cycle because it's very expensive. So behind the technology is not only in some of the countries to solve the problem of the shortage of embryologists, which is not the case of Spain because we have a lot of embryologists here, but is maybe also to reduce the price or the cost of the cycle and make affordable for more people. And then that could be the reason of the growing in the number of cycles in the next years. If we are able with the technology to make the IVF more affordable and then accessible to more patients, which will be the reason behind this growing and the way to being able to absorb also this increase that we expect. Actually, if you talk with many specialists or even the venture capital that want to invest money in this field, they believe that there's going to be an increase in the number of cycles in the next years. And for many reasons, maybe for social reasons, the way that we're going to reproduce in the next year, the new generations. But in any case, in order to be able to afford this growth, we will need to use technology to do that for many reasons. Some of them also to increase to reduce the cost and also to be able to do more cycles for sure. There is one question related with other things about your culture that maybe is interesting to mention. It's about the intravaginal culture. Because at the end is, I mean, you know that there has been several pilot studies and studies done about this. What is your opinion about these devices and if they have future, maybe also for reduction of the cost of the cycle, for example?
Yeah, I mean, I think none of those studies that have been published have shown a benefit over conventional systems. They've not been superior or even equivalent in many cases. But I do think that, again, going to developing countries where they don't have access to full laboratories, then it's a viable system. It is a viable system, but it's, as they say, it's horses for courses. And I don't actually see vaginal culture actually being a main technology that we use in IVF, no.
And the other thing that is, I mean, it's coming to my mind and my memories is about the endometrial cells and tubal cells. You remember, I mean, my PhD director, Carlos Simon, and all the work that he did with co-culture. You are good friends too. How is, I mean, how is the interaction with these cells and if why we're not using now, I mean, the reasons behind that I can understand, but can you give us a comment about that, about endometrial and tubal cells?
Absolutely. You know, we did play with co-culture quite a lot ourselves, and I had the pleasure of having Fulvio Gandolfi spend a year with us when we were in Monash back in the early 90s, and Fulvio was one of the pioneers of co-culture in cattle. And we developed basically a sequential system for cattle embryos, and it outperformed the co-culture. So, you know, that was kind of around that time around the world, we saw the demise of co-culture. Sequential media showed that it actually gave better than the co-culture. Here's the catch. The co-culture cells were somatic cells on a plastic dish. You know, it was 20 odd years ago we were trying to grow these cells. Now, fast forward to 2022, where we're talking about growing cells on 3D scaffolds. So I have thought about this, Marcus. I think it's a really good question. What if we use 3D printing devices, which I alluded to on one of the slides, where you could actually create a scaffold, that we could actually have proper three-dimensional cultures of fallopian tube and or endometrial. So truly recreating an in vitro female tract. That would be fascinating. And I think that would be a great PhD students project, or a whole team of students projects for many years. So we should probably talk after this about that. But yeah, I think that holds a really intriguing question. But again, that's, Mina, that's definitely one for you 20 years, not 10. Yeah.
Right. There is a question here from Jeremy Thompson from Adelaide.
I've heard of him.
Yeah, the pathway to automation of IVF is not a tomorrow event. It will occur iteratively and should be done with a view of making the lives of embryologists easier and better. What are the panel's thoughts? Lorena?
Yes. I also think that the automation is not something that it's going to happen now for all the tasks, but I said before there are some tasks that it's expected that in the near future we will be automated, and it's easier to automate, for example, functions for data collection or oocyte denudation, maybe fertilization checks with image analysis. But I think there are other procedures more complex, such as ICSI or trophectoderm biopsy or vitrification, that maybe they're not be fully automated in the near future.
Yes, it looks that the automation is coming more from the software than hardware. I mean, automation can be definitely observed now in some of the tasks that can be done by computers, but in the handmade side, that's quite much more complicated, and there is more room to do. Even that, also I would like to say that make the life of embryologists easier, you have to ask this question into the lab. Today, if I go to the lab in Valencia and I ask this question to my colleagues, they don't believe that, because they would like to keep working as embryologists like they do now. They love to do denudation, ICSI, vitrification. They don't want to be substituted by the machine to do other stuff. I will say that a big proportion of the embryologists are not really interested in research, which I understand. They like their work in the lab. They like to study, they like to see new things, but they like this handwork, and they don't want to have more time to other tasks. That's my feeling. If you ask too many embryologists, I'm pretty sure that the rest of the panelists, Romualdo, that is working in Edinburgh, that believes the same, that most embryologists like to do the work in the lab, right?
Yeah. Well, actually, I'm agree. Me personally, but many I've got. Yeah, I completely agree with you, because I mean, I personally enjoy still doing ICSI, practical work, vitrification, and I still have been. I mean, every day. Although I'm doing ICSI for decades, but you know, I have the curiosity to come back the day later and check the PM, because you know, to see is. Yeah, I mean, but I guess that my opinion is maybe automation can be used or applied in maybe in very large laboratory. I mean, even vitrification. I mean, some vitrification in automatic vitrification more or less is already there, but there are some kind of complication, and I mean, an embryologist still have to be there. So if you are a relatively small lab and you perform a few cycles of vitrification, you are not going to invest in big money to do it, but you have your staff and you prefer doing by an embryologist. That's the and then there is a point, as you say, that some kind of you know, we are speaking microfluid and many other technology for decades, but you know, to me, it's still some time that they will come really applied in an affordable way as well, because there's very often some kind of you know, investment, big investment as well. So not every lab they can reapply for that.
Just one if I could ask one last question of oh, sorry, David.
No, I was just going to add, I agree with what the guys have said, but I also think that people don't realize that their jobs could be made easier and less stressful. And we give ICSI as an example. I mean, that's, if you've got a lot, 20 egg ICSI, and you've got to do 5 of them in the afternoon, you're going to feel it at the end of the day. If automation can help you do those ICSIs, it might not completely alleviate all of the things, but all of a sudden your life is less stressful. And I think we're going to see a lot less recurrent injuries to embryologists as some of these technologies come in. But it's like all technology, you don't know how it's going to impact you till you have it in your hands. And give an example, you know, a smartphone. You know, look how that's transformed everything. Everyone was very dubious when they first came out. Now you wouldn't leave the home without it. In fact, you don't. So I think there's a degree of, yeah, we love our jobs, we love doing what we do, but until you see what could be done, you don't really have the full picture to reevaluate, oh, actually, my job's still really cool, but this part of it is a lot less stressful now because I have a device that can help me do it. I never see a day when there aren't lots of embryologists in a lab. That's for sure.
Romualdo, there was a question for you about preventing collapse. I wonder if you have any thoughts on that?
Preventing, I mean, this kind of collapse are what can be defined as a natural collapse, as a spontaneous collapse. So, I mean, since we've seen these specifically using time-lapse monitoring, so we do not think they are related to some kind, because I remember even a long time ago, some people that were saying maybe it's a kind of change of temperature. When you take the dish out, this kind of stress can be related and induce this collapse. But I mean, this one that we like to define as a spontaneous collapse. So I don't think they are induced by any alteration from an operator. So I think since they are a kind of natural, I don't really think they can be prevented. Actually, it would be nice more to investigate. And there is actually a recent paper I think I read from Peru, in which they compare. I mean, they've done some genetic analysis and they found that the blastocysts, they show 1 of 2 collapses. They have high aneuploid rate, which is, you know, compared to embryo that do not show collapse. So maybe that will be something to, you know, to investigate further in the next future and see really probably there is some alteration in genetic or in aneuploidy that might be related to this event. But definitely I don't think it can be avoided, you know, in terms of something can be done in a different way. No, I don't think so.
Thank you so much. I hate to bring this to bring this to a close, but I think we are now close to 5 p.m. Eastern Standard Time. I think it's time to wrap it up.
It's time to finish. It's time to close this session. I mean, here in Spain, it's close to 11 p.m. So it's time to go to sleep.
To bed.
Tomorrow we have to go to the lab to do again ICSIs, vitrification, and things like that. Unfortunately, because the machines are still not working. And first of all, well, from my side, I would like to thank, I mean, all the people that has attended this RBM online live webinar, our editor, Mark Fauser, Mina, of course, Richard, the panelists, David Gardner, because it has been an honor and a pleasure to have you here. We feel really, really lucky to see your presentation and your thoughts. You are amazing. I am one of your number 1 fan. the future embryologists that, well, not Romualdo maybe is not, but the future embryologists like Lorena, the view also of Ana Pereira from another perspective of the embryo. So we have all these views in front of us. It has been very interesting. Also, I would like to thank the organizing committee, the I3 initiative, with Jax Cohen, with Peter Nagy, with Marianne. I'm not going to pronounce well your surname because I'm Spanish. Sasha Bastes. If I do wrong, I'm sorry. And Jill Spammer, of course, and Thomas Elliott. And I give the final word to Mina to close this session.
Thank you so much, and thank you everybody for joining us today. Please join us again for the next RBMO live episode on the 7th of June with Professor Nick Macklin, who will be showcasing the latest developments from our reproduction technology and society section. That really promises to be another great session. Before then, come back to I3 for a new topic on the 22nd of March. It's called, I think I read it somewhere. I think Giles comes up with these titles. They're just, you can't beat them. All about leading and influential papers in ART, the backstory and circumstances around some of the game-changing articles that shape IVF today. With a great lineup: Peter Nash, Colin Howes, Jacques Cohen, Kathy Go, Duncan Nicholas, and David Gardner again. David, did you know that?
It's a cameo appearance, I think.
Okay, it's a cameo appearance. All right. Thank you so much, everybody, again, and we'll see you soon. Take care. Bye-bye.
Great talk to everybody, and thank you to the
all.
audience for your interaction.
Bye.
Lovely time.
Bye.
Thank you very much.